Hello everyone, welcome to today's National Soil Survey Center webinar. My name is Sean McVeigh, National Training Coordinator for the Soil Science Division and host for today's webinar, Microbial Diversity of Oregon Soils, Links to Soil Health. This webinar is being recorded and all participants join the webinar in listen-only mode. You receive the webinar audio through your device's speakers. There isn't a telephone dial-in. If you're having audio difficulties, please check the various ways your computer speakers may be muted or have their volume set low, including the speaker adjustments available in the Adobe Connect interface. You can maximize your webinar experience at Adobe Connect by shutting down VPN and any other programs that might compete for bandwidth. This includes email and Microsoft Outlook and instant messaging and Skype. Taking a look at our webinar room layout, Adobe Connect has content pods that include the feature presentation, chat, and Q&A pods. Use the 4-arrow icon in the featured presentation pod to enter and exit the full screen view as you choose. To submit a comment or question for me or our presenter, use the Q&A pod and type in your question. I'll handle technical difficulties the best I can while hosting the webinar and interact with our presenters to answer your questions during verbal Q&A periods. Today's speakers are Dr. David Myrold and Chris Burgess. David Myrold is a professor of soil microbiology in the Department of Crop and Soil Science at Oregon State University in Corvallis, Oregon. He earned his PhD in microbiology at Michigan State University in 1984 and MS in soil science from Washington State University in 1979 and a Bachelor's of Science in Forestry from Michigan Technological University in 1977. During his research career, he has studied most of the major processes in the soil nitrogen cycle. In the last decade, he has investigated the connection between the composition of bacterial and fungal communities as it relates to soil nitrogen and carbon cycling. He has worked in both forested and agricultural ecosystems. Chris Burgess earned his bachelor's from the University of Maryland in 2011. He joined our research group as a PhD student 3 years ago when he began working on the project that we are going to describe to you today. The webinar over to you.
Okay. Thanks for hosting the webinar. As Sean said, I'm Dave Myrold, and Chris Burgess is with me here. And I'm going to start off with an overview of the project and present some background about the approach we use to characterize microbial communities in Oregon soils. Chris will then take over and review some of our findings about the bacterial and fungal communities in the soils we studied, and then I'll finish up with the linkages we've found so far between microbial communities and soil health. Next. Okay, so I'll begin with a little history. Hopefully you see this interesting image of Oregon with some of the soils lined up inside the outline. So a few years ago, the fall of 2013, I had a master's student who finished up with me, Megan McGinnis, and she started working as a volunteer with Corey Owens, who was working out of the NRCS office in Oregon City at that time doing soil health assessments. And as they were talking, Megan mentioned her research about the effect of forest harvest on soil microbial communities and activities. And this prompted Corey and then Thor Thorson, who was the state soil scientist for Oregon at the time, to contact me about doing some research on the microbial communities in Oregon soils to see if there might be a connection with soil health. This ultimately led to a proposal that was funded by the NRCS and managed by Skye Wills out of the Lincoln Lab. It was officially called Integrating Soil Microbial Community Composition into Soil Survey as an Index of Soil Health. But we refer to it as the Microbe Map project. And in this Microbe Map project, one of the goals was to integrate the microbial community data that we collected with the existing soil survey data information on soil physical and chemical properties that's housed in NACIS. And then the second one was to explore the relationship between soil health and microbial communities. Next slide. Okay. So hopefully you're seeing a very colorful map of the common resource areas in the state of Oregon. And the study design was really influenced by the help of Thor Thorson and other NRCS soil scientists who were working out of the Portland office. Based on his knowledge of the soils of Oregon, Thor stratified soils by CRA to capture the diversity of the soil types and land use present within the state. This included soil series that occupied large land areas and also representing unique features. So we wanted to make sure we captured all the soil orders. We had a serpentinitic soil that was present there as well. Then in the spring of 2015, with the help of a large number of NRCS soil scientists, a few of whom I think are on the call, and those from other agencies, we were able to sample 113 sites throughout the state of Oregon. At each location, a complete soil profile description was done. We made an in-field assessment of soil health, and we collected 5 separate soil samples from 0 to 5 centimeters, or 0 to 15 centimeters in depth. Shouldn't have put me in control of the arrows. I can't get them right. Okay. So this map of Oregon shows the 113 sampling sites which we had, and these included forests and rangelands, pastures, as well as croplands. We have 4 land use types. The symbol colors represent the 10 soil orders that were sampled, and then by each one of those sites there's a tag which lists the name of the soil series for those of you who might be familiar with some of those soil series. Across the state, the dominant soil order were mollisols, making up almost half of the soils that we sampled. But we did have more than 10 soils that represented the andosol, iridosol, and then septisol orders. Because we had these 5 samples within each site that allowed us to capture the heterogeneity within a site, we analyzed in total 565 soil samples. So I'd like to give you just a little bit of a background on how we took a look at soil diversity. And so I'm going to start with the basics, with the tree of life, which contains 3 different domains, the Archaea, the bacteria, and the eukarya. Now the eukarya are, of course, the ones that we're most familiar with. That's where the plants and the animals are. And we often think of the huge diversity above ground with plant, animal, and insect life. But all of that actually fits just into a few twigs on the eukarya branch. One of those twigs includes the fungi, which is something we measured in our sample. And the fungal diversity is probably as great as that of the animals, plants, and insects all combined. And so then if you look out to the Archaea, which contain many more twigs, and the bacteria, which contain even more, it gives you a sense of the huge amount of diversity of life which is present. And members of the Archaea, the bacteria, as well as the fungi and some of the soil animals are all found present in soil. Because Oregon has such a wide diversity of soils, we have 10 of the 12 soil orders, we would expect that the microbial communities, as you go across the different landscapes and land uses in the state, would be quite high. But in addition to that, even at the small scale, soils are inherently complex environments. So that when you get down to the level of an aggregate or maybe a teaspoon of soil, we find that there's 1 billion bacteria, and that could involve 10,000 different species of bacteria. That same gram of soil probably holds up to 100 meters of fungal hyphae, and there might be 500 different species of bacteria. And of course, there's numerous soil animals as well. So there's a lot of diversity out there, and we wanted to capture that in the method that we chose to assess this microbial community. Okay. So there's a number of different ways that you can assess the microbial community. Microbial lipids have been used for more than 30 years. This includes methods you may have heard about. PLFA and FAME are the common abbreviation for those. And they do a good job of providing an index of the total microbial biomass, as well as providing information on both bacteria and fungi, but they give very limited taxonomic resolution. The procedure for the lipids involves extracting lipids out of soil using an organic solvent and then subsequently analyzing those with a gas chromatograph. There are some commercial labs which are doing this analysis at the time. Another method is to look at the DNA through a method called DNA amplicon sequencing. This focuses on a particular marker gene, and it provides great taxonomic resolution, far greater than what you can get with the microbial lipids. And it's really become the current standard for doing microbial community research. It involves extracting DNA from soil, amplifying a gene of interest using the polymerase chain reaction, and then sequencing these amplicons. Another technique that's still very much in the research stage is shotgun metagenomics, which you can think of as sampling or sequencing all of the genes of all of the organisms in a given sample. But that still has not been applied in a widespread way to soil. So we chose to use DNA amplicon sequencing. And the molecule you've been looking at is actually the 16S rRNA gene. It's very useful in terms of looking at the diversity of the bacteria and the fungal communities. Its sequence tracks the evolutionary processes, so we can put it into the sequences of the tree of life like you saw on the previous slide. And there are extensive databases that allow us to take a DNA sequence and match it up with taxonomic information. The same holds true for this ITS gene, which is used for fungal sequencing. And altogether, with the sequencing technology that's available, you can get more than 15 million sequences in a given run. You can sequence both the 16S and the ITS in the same run, and you can do multiple samples on a run. You can multiply up to 100 samples, for example. So that makes it very efficient and cost-effective. All the sequencing that was done in our project was done by the Earth Microbiome Project using facilities at Pacific Northwest National Lab and Argonne National Laboratories. So when we take a look at the microbial data, we have literally millions of sequences, and so following quality control and making sure those are all contain good information, we have grouped them at a level of 97% similarity. This reduces the total number of pieces of data that we look at, but it still gives us a lot of resolution. This 97% threshold is roughly at the genus level, so fairly specific. We call those operational taxonomic units, or OTUs. If we take a look at our data set, the bacteria in Archaea, we had a total of more than 3 million sequences, which ended up giving us 23,000 different OTUs, or bacterial genera, if you will. In a given soil sample, on average, we had more than 2,000 different OTUs, which gives us quite good coverage of the soil. In the case of the fungi, we had about 4 million sequences, 9,000 different OTUs, and in a given soil sample, about 400 different OTUs, which does a great job of covering the fungal diversity. So with that as background, I'm going to turn it over to Chris, who's going to share some of the data on the microbial communities of Oregon soil.
All right. So as Dave previously highlighted, there's a large amount of microbial diversity within our study. But we also have a large range of soil types. So within the 10 soil orders that we sampled, we have 68 different subgroups represented. So with this much complexity in both our soils and our microbial data, it makes it sense to take a zoomed out view of the data and look at it from like a very high level view, which we did here. So in this graph, it shows the bacterial communities for each of the 10 soil orders. Each of these horizontal bars represents a soil order, and the colors represent the 11 most dominant bacteriophila that we found in our study, with the width of the color showing how prevalent that bacteriophilum was in that particular order. So for example, at the top we have alpha-sols, and our 2 most dominant bacteriophila in that order were proteobacteria and acidobacteria, the teal and then the pale yellow. It's worth noting that these dominant bacteriophila are what we typically find in soils. And so for the most part, proteobacteria and acidobacteria are the 2 most dominant phyla that we found in the study. However, this isn't always the case. So if we look at iridosols, the 3rd bar from the top, we see that it has a large proportion of actinobacteria, that dark blue color, compared to the acidobacteria. So actinobacteria are typically associated with a dry,
high pH soils, while acidobacteria are associated with our lower pH soils, which makes perfect sense. At least for Oregon, our aerisols typically have a high pH. But comparisons at this resolution can only take us so far, and if we really want to get an idea how these microbial communities are distributed across the state, we need to dig a little deeper. So instead of looking at the bacterial communities from the phylum level, we can zoom in and look at the microbial data from the OTU level. But there's just one small caveat. So if we wanted to represent, so we represented the 11 most dominant bacterial phyla with 11 different colors here in this graph. But if we wanted to represent the OTUs, we would need over 22,000 different colors, which just isn't practical. So we need some sort of technique that can take this high-dimensional microbial data set and distill it down into information or useful information that we can use. One of these techniques is called ordination, which is this is the plot of. So all an ordination is it takes multidimensional data and tries to distill it down into a smaller number of dimensions while still preserving the shape of the data. So it does this by finding directions or axes that represent the most variation that we see in our data. So in this particular ordination, we see that our first axis, our x-axis, captures about 20% of the variation we saw in our bacterial community data, while the y-axis represents about 8%. So how do you interpret a graph like this? Well, each of these points on the graph represents the microbial community at a given sampling location, and I went ahead and colored them by their soil order. Their x and y coordinates isn't entirely important. What is important is the position of the point relative to all the other points. So this is because points that are closer together have similar microbial communities than points that are further apart. And so ideally what we would want to see is some sort of grouping or clustering based on the coloring that I'm using here. So, to get an idea of this grouping of soil orders, and we don't, at first glance, we don't really see much of a pattern, but if I go ahead and highlight some of these soil orders like I did for the aerisols, we see that we have a that all the aerisols are all pretty much grouped together on this right-hand side of the graph. And if I look at it at the alpha sols here, though they are vertically distributed across this ordination, they're all pretty much constrained on this right-hand side, leading us to believe that there may be some sort of information in the way we do soil classification that helps us explain or bin our microbial community. Well, how well does that bin it? So in this slide, we have the same ordination on the right-hand side, but on the left-hand side, we have the results from a statistical test called PermaNova, which is a multivariate ANOVA. So you give it my microbial data, and you give it a way that you want to classify or group the data. So the groups that I used were land use, soil order, and suborder. And given that, it tells you how much variance is explained given that grouping. So all of these tests came back statistically significant. And what we see here is that soil order and land use both explain similar amounts of variation. But when we move up to the suborder level, we see about a 50% increase in the variance explained, leading us to believe that there is indeed some facts about how we classify our soils that helps us explain bacterial communities. The problem is that we can't really go at a finer taxonomic resolution from the soils because we don't really have the replication within the study at the great group or the subgroup level to look at it from a PermaNova aspect. It just wasn't the way the study was designed. But what we can do is we can look at the physical and chemical characteristics of the soils themselves and see how those factors are influencing how our microbial community is distributed. So here I have the exact same ordination, except instead of coloring it by soil order, I've colored it by their pH. And immediately a pattern should jump out. We have high pH soils on our left-hand side, and then as you move right, the pH drops or gets more red. So there is a very horizontal pattern here, but we don't really see that pHs really explaining much of this vertical variation that we see in our ordination. So another way to represent this is we can calculate the correlation that pH has between our x and our y coordinate and then plot it in as a vector. So the direction of the vector tells us where the strength of, or what the influence is on that factor on that particular axis. So we see that soil pH is really only influencing our x-axis or helping explain our x-axis variation, and then the length of that vector tells us how strong that correlation was. And we do see, in fact, that, yeah, it's only explaining horizontal variation. So what about this vertical variation? Well, we can go ahead and plot all the other factors that we have measured in our data set so far. And we see, so soil pH and mean annual precipitation are exactly almost pointing directly horizontal, and which makes perfect sense in Oregon because on the coast at least we have our low pH wet soils, but as we move inland we have our high pH and dry soil. So for Oregon, these two factors are highly correlated with each other. But if we look at some of these other factors that are maybe pointing vertically, we see that maybe the texture, which is quantified in our standing clay percentage, and mean annual temperature, may be helping explain some of these other variation, some of this other variation that we see in our microbial data. So in essence, I think a lot of our bacterial data is explained through these physical and environmental factors that we see. So what about the fungi? Well, so if you remember from the very first plot that I showed you about the bacterial data, this is the exact same thing, except instead of the major bacterial phylum, I'm showing you the major fungal phylum. And if you remember the bacterial phylum, the major difference between this one, I feel, is that there's a lot more variation that we see between soil orders in the fungal communities. So leading us to believe that maybe soil order, at least for helping describe the fungal phylum, may not be as useful as it was for the bacterial data. If we look at spotosols and ultrasols, the two soil orders close to the bottom, we see that they have a large proportion of this teal color, which is basidiomycota. And that's been typically associated with wood degradation and ectomycorrhizal. And so, at least, well, spotosols and ultrasols are also where you would find our forested, wet forested sites. And so, maybe leading us to believe that vegetation may actually be an important driver in helping us describe fungal communities. So, this is, on the right-hand side, we have the ordination of the fungal data colored by soil order. But on the left-hand side, where I want to focus right now, is where we have the Permanova results for our fungal data. And what you see similar results that we do from the bacteria. We see soil order and land use both explain similar amounts of variation, but they explain less variation compared to the bacterial. So the bacterial explain about 20% of the variation, while the soil order, so the fungi only explain about 15%. But if we move up to the suborder level, we see we get about 100% increase in variation variance explained instead of just a 50% increase, making us think that, well, at the soil order level, we may be missing something that helps us distinguish our fungal communities. But when we move up to the suborder level, maybe there is something at the suborder level distinction that helps us explain our fungal communities. Well, what may these factors be? So I went ahead and plot the factors on the ordination for the fungal community. And so we see aridity and soil pH are pointing down to the bottom left-hand corner, and they're pointing at a lot of our iridosols, which are colored in that kind of olive green color. And we have mean annual temperature almost pointing vertically. But what I wanted to highlight is we don't really see any of these factors kind of pointing horizontally or helping us explain much of this horizontal variation. We see some of these vectors have a horizontal component, but none of them really point horizontally. So what may be partitioning out this horizontal variation? Well, if we look closely, we see that there are potentially maybe two different clusters of points. We see on our left-hand side, we have a large kind of cluster that spans the whole width of the graph. But if we on our right-hand side, we see this smaller cluster of points. And if we look closely, we see on this right-hand side cluster, we see that it's composed of inceptosols, spotosols, ultisols, and andosols. These making me think, well, maybe there is a land use component here or a vegetation component that is partitioning out at least our x-axis. And so what I did was I colored them by land use instead of soil order, and we see that, in fact, that, yeah, almost all of our forested sites are contained in this little cluster on our right-hand side, making us believe that vegetation is a very strong selector for our fungal community. So for conclusions of this part is we found evidence that using soil classification or soil taxonomy and helping us describe soil bacterial community patterns might actually be useful and worth pursuing. Bacterial communities, at least in our studies, seem to be generally governed by their chemical and physical characteristics of the soils themselves, while in our fungal communities we see a stronger influence of vegetation selecting on the community over these physical and chemical properties. And with that, I'll pass it back over to Dave.
Okay, thanks, Chris. So to begin talking about the connection that we have been examining between soil health and the composition of the bacterial and fungal communities, I'm going to first start off with a little bit of a overview of soil health assessments. And what is listed on this slide are three different assessment systems, if you will. The Comprehensive Assessment of Soil Health, that's sometimes known as the Cornell Soil Health Test. The Soil Health Institute has their Tier 1 measures. And then there was the Soil Management Assessment Framework. I picked some of the measures out of that on the rightmost column. All of the tests, if you will, that are in regular font are the minimum data set for each of these three systems. The italicized methods are kind of like add-ons that can be used. Okay. And you'll notice that above that space that I have there, most of these methods are in common across. So they all have some measure of available soil water. They all have some measure of water-stable aggregates or of organic matter. But it's only one or two characteristics out of those lists that relate at all to the biology or the microbiology of the system, respiration or nitrogen mineralization. And both of those not only are a function of the microorganism, but they're also a function of the amount of carbon, the type of carbon, amount of nitrogen, type of nitrogen that's present in the soil. So they're rather indirect measures of the biology, if you will. Now, in the SMAF set, they do have a couple of more direct measures that are included in their optional list, microbial biomass or a nematode index. And so from my perspective, this raises the question, if soil health is really centered on the living soil ecosystem, why aren't there direct measures of the biological component of soil that are incorporated into these different systems for assessing soil health? And that's a big part of what we're hoping to pursue with the project that we have. So if we want to think about what biological characteristics to consider, there are at least three that come to mind that relate to the microbial community. The first is the abundance, or just how many microbes are there, what's the population size of the bacteria and the fungi, for example. The second is diversity, and that relates to the number of different types of bacteria or fungi that might be present in the soil. And the third one is composition. And composition is what different types happen to be present in a given soil. And collectively, they all influence the functioning of the soil microbial community. So in the recent years, a couple of years ago, there were some meta-analyses done on what we know from soil systems with respect to how abundance, diversity, and composition influence soil function. So in this review of the connection between abundance and function, most studies have found a positive relationship, especially for specific functions. In other words, the more of an organism that does a particular function, the higher the rate of that function. A good example there is nitrification. The more nitrifying bacteria that are present, the higher the rate of nitrification. In the case of diversity, this connection has given much more mixed results. For example, reviewing all the studies suggests that a lower diversity results in lower soil respiration, yet there was no effective diversity on litter decomposition. And we often would think of litter decomposition and soil respiration as being pretty similar in terms of microbial function. And lastly, with respect to composition, most studies that have looked at the composition of the microbial communities, what different types of bacteria or fungi are present, have tended to find that the functions being measured have varied with those communities. So collectively, this suggests that potentially there is a pretty good connection between direct measurements of the microbial communities in soil and how they're functioning. So in our case, we assess soil health using an in-field assessment. And it's probably too small for you to read there, but it's basically a scorecard that was developed by Corey Owen, and it was split into our 4 land uses, so croplands, pasture lands, rangelands, and forest lands. And zeroing in on the croplands, which is what I'm going to be showing you data for, in this cropland assessment, there were 7 attributes that we rated while we were out in the field. Soil structure and compaction were the first 2. Then there was an assessment of whether the soil seemed to contain a lot of organisms. In particular, the presence of earthworms was noted. Was there plant residue? Did this look like it was actively decomposing? What was the status of the plants which were growing on the site? Did they look healthy and vigorous? And finally, what sort of root growth did we observe? Do we find roots growing throughout the soil? Was there some sort of a restrictive area? Those sorts of characteristics. And so each one of these 7 attributes were rated on a scale from 1 to 10, and then we pulled all of these together. And so for each land use, we basically normalized the scores that we got and expressed them as a percentage, and you can see that in the bar charts here. And for each of the land use, we got a fairly large variation in the soil quality score, from scores that were relatively low to scores that were quite high. You know, the mean of these varied from system to system, which is probably as much a function of the scoring table or scorecard that we used as the particular land use type. And if we, within each land use, if we split them into the top, middle, and bottom third, you can kind of see the different colors there. And so in the case of cropland soils, about a third of the soil scored below a score of 50. About a third scored roughly about 70, 75 or above, and the rest were in the middle. So we got a pretty good spread of soil quality scores, which means that it should be quite good to relate those to the microbial communities. If all the scores turned out to be the same, then it'd be pretty difficult to see if there was a relationship between the microbial communities and soil health. So focusing on the cropland sites, here's a map that illustrates those cropland sites that were sampled. There's a total of 38 of them, As you can tell from the blue color, most of those were mollisols, but we did have 4 iridosols, 2 alphasols and andisols, and then we had an endsol and a septisol and an ultisol as well. And as you might expect, they're distributed around the state of Oregon in relationship to where the major agricultural production areas are, at least cropland production areas. So these were the soils that we used in our analysis. And we examined a couple of different measures of microbial diversity. The first is alpha diversity. Okay? And this is more the diversity within site. And it's often thought of as richness or evenness. So by richness, I mean the number of different species or types, or in our case, OTUs that we measured. And the little cartoon on the right kind of illustrates this. The ellipse on the right side has more different types. I guess those are supposed to be trees than the one on the left. And so a higher richness means more different types of species in the sample. Evenness has to do with whether the relative abundance of those different taxa are similar. So on the right-hand side, we have a very even distribution of the species. There's 2 of each. On the left-hand side, we have a relatively uneven distribution. Basically, there's a couple perhaps dominant taxa and a lot of rare taxa. Now, sometimes these concepts of richness and evenness are combined into indices. The Shannon Diversity Index is one example of those. Okay. So if we turn now to the data, looking at bacterial diversity, we have 2 plots here. The left-hand side is the richness, and next to that is the Shannon diversity. The soil quality score is plotted along the bottom, and the data points are colored pink for poor and green for good. And you'll notice a couple of different things. First, there's quite a bit of scatter, quite a bit of variation within the data. And this probably isn't surprising, considering that we have soils from 7 different soil orders and from regions from across the state. They're growing a variety of different crops, and they're under different management systems. Nevertheless, there's a general upward trend with higher soil quality scores being associated with greater alpha diversity, whether that's richness or the Shannon index. And this is a little bit more clearly shown if we plot the soil quality score as a function of bacterial diversity. In this case, it's the Shannon index, which is plotted on the x-axis. And here we took the mean diversity for a given site. So there should be 38 points up there. And what you can see is that there's a positive trend, that generally the greater the bacterial diversity, the higher the soil quality score. We can do the same sort of analysis with fungi. And so again, richness is on the left and the Shannon index is on the right. And we see a somewhat different pattern. As you go from the lower quality to moderate quality sites, there's an increase in fungal diversity, which we might expect, but actually the best sites actually have a slightly lower diversity. And so it's interesting to speculate why this might be. We would perhaps expect that diversity would increase with soil quality score, but eventually it would level off. But here we see this decrease in the highest soil quality scores. It could be that at this highest level, what we actually have is a few highly beneficial fungi who become dominant in the system, thereby increasing soil function or the soil quality score, but the overall diversity of the fungi is actually a little bit lower. In order to perhaps get a better idea about this, we can look at a second aspect of diversity, beta diversity, which is something Chris has already introduced, or how the microbial communities vary across different soils or among soils. So this is an ordination plot. This is an ordination plot of the fungal community, just for those cropland sites. And here it's been color-coded with the lighter blue colors being higher soil quality, the darker blue colors being lower soil quality. And if you take a look at it, you can see that by and large, the lower quality soils tend to group in the upper half of this diagram. The higher quality soils seem to group more in the lower half of this diagram. Now, I also plotted those vectors which Chris introduced you to, and as with his ordinations, aridity, or mean annual precipitation, is a real strong driver of separating sites on the left from the right-hand side of the diagram. And so on the left-hand side of the diagram are Western Oregon sites. On the right-hand side of the diagram are Eastern Oregon sites. And so if you kind of think of it as Western Oregon, Eastern Oregon, within each of those clusters, you still see this pattern of higher quality scores associated with being lower down in this ordination. So it suggests, well, and I guess one other thing I should point out here is that still the Western Oregon soils are quite separated, microbial and fungal communities in those soils are quite separated from those in Eastern Oregon. So there seem to be different, if you will, beneficial fungi in Western Oregon soils compared to Eastern Oregon soils. This is the same type of an ordination, but in this case with the bacterial community. And so there are some similarities, but it's a little bit messier ordination. And by that I mean we don't have quite perhaps as distinct a separation of the soil quality scores with the microbial communities. So if we think of it with this east-west split again, if we look at the eastern side of the state, we do see, generally speaking, that those soil quality scores for those bacterial communities is a little bit lower. But within that cluster of lower quality scores, there's a few locations where we actually have quite high soil quality. And then if you turn to the western side, most of those bacterial communities are associated with soils of relatively high soil quality, but embedded within that cloud of light blue dots are some sites which have relatively poor soil quality. Now, we don't know exactly yet what the reason for that is, but it's going to be interesting to take a look at those sites in greater detail, because perhaps the poor quality sites amongst the sea of higher quality sites represent some systems that are under a different management regime, or perhaps they have a different crop site associated with them. So I'd like now at the end to summarize some of the insights that we got about soil health and their relationship to microbial communities and some of the next steps. So we did indeed find that there were relationships between the bacterial and fungal communities and their diversity or their composition of those communities with respect to soil health scores. The pattern for diversity seemed to be stronger for bacteria, although we have a possible explanation for why it wasn't quite that way for fungi. And in the case of fungi, there seemed to be clear differences in the composition of those communities and how that was related to this in-field soil quality measure that we made. Now, we still have additional analyses to do. We're in the midst of generating data so that we can put together a soil health score based on some of these laboratory-based metrics that I introduced at the beginning, like in the Cornell soil health test. We will be generating data to look at the abundance of bacteria and fungi to see how those relate to these soil health scores. And then we have the ability, using the sequencing approach, to see if there are particular bacterial or fungal taxa which are associated either with higher or lower soils with either higher or lower soil quality. And as we look to the future, there's a couple of things which I think are going to be important that needs to be done in terms of really deciding whether this microbial community information is useful from a soil health perspective. First, we need a greater density of sampling within selected sites. As Chris mentioned, we have some interesting clues that there may be this relationship between the composition of microbial communities and the taxonomy that's used in soil classification. Well, we need to be able to sample more soils within a selected region because we'd expect these soil health measurements to have some region specificity to it. And secondly, our study was not designed specifically to test the effect of different cropping systems or management systems on the health of soils. And so I think we need to do some studies where we specifically look at different management systems and see what influence that may have on soil health metrics and see if that's reflected in differences in the microbial community. The work that we've talked about today involves lots and lots of people, and I've listed some folks from OSU, some of the landowners, the people who helped us out with the sequencing, and then I think I have a comprehensive list of all of the different NRCS scientists as well as some other collaborators who helped us out with soil sampling, the soil descriptions, and the other information. So I'll leave this slide up, but I think we're open for questions at the moment. Thank you for your attention.
All right, thanks, David and Chris. And while we give people a moment to compose their thoughts and put a question in the Q&A, I'll just go ahead and start with one. Do the microbial populations change throughout the year? And I guess I missed earlier on, you know, over how many months were the samples collected? Was it the same time of year, or was it sort of spread out across the whole field season time frame?
Yeah, so there's this whole other aspect of the project that we didn't talk about, but most of the sample, all the data that you saw today was sampled in spring of 2015. But we also sampled a select few sites every month for an entire year, which folks in Tillamook and Pendleton helped us sample that. And what we found there is that we found, even though they were sampled throughout the month, throughout the year, we still saw a very strong spatial selection on these microbial communities, indicating that they do change throughout the year, but the spatial variation of the data seem to be driving a larger difference between our microbial communities.
And I'll just add to that, we had 3 soils near Corvallis, which were probably within a kilometer or 2 of one another, and they happened to be on different soil series. And even with that monthly sampling, you could tell the microbial communities of those 3 soil series apart from one another. So that spatial variation overrode the temporal variation.
Interesting. We do have some questions coming in. So we've got one from Charles. In your opinion, what is the impact if microbial populations on organisms, for example, amphibians? Could these species benefit from a healthy, non-pathogenic microbial population?
Okay, well, I'll take that since I'm still here. Yes, I mean, it could be in a very direct sense, as you mentioned, pathogens, some of which might exist or be able to survive in the soil system. But I'm thinking it's probably more through, I don't know if you want to call it quite a food chain effect, but if you have the microbial community contributing to greater plant vigor and higher above-ground productivity, that presumably would enhance the population levels of the organisms that feed on those, and maybe, you know, and so it's kind of related in that sense, maybe to an overall picture of ecosystem health, as opposed to a direct effect on, I think you mentioned, amphibian.
All right, Travis has a question. Wants to know if he saw this correctly. On considering if population was good, fair, or poor by land use, it looks like each land use had a different threshold that you used to make the groupings. Is that correct?
Yeah, that is correct. So using the assessment and trying to break these assessments down into a score between 1 and 100, the scores didn't break down roughly evenly or normally distributed. So what I ended up doing was letting the data guide how I bin these soils between good, bad, and poor by just binning the top third in good and the middle third in moderate and then the bottom third in poor, depending on the land use itself. Yeah.
And the attributes were very different for each of those land uses, so you can't, it's kind of difficult to compare a 60 in the cropland with a 60 in the forest, for example. I mean, to be quite honest, I was very surprised that this relatively I'll say, semi-quantitative field assessment that we had actually correlated as well as it did with some of these microbial community measures. And so I really look forward to some of these more quantitative lab measurements that are used for a soil health score to see how those play out.
And Brian has a question, has there been any success in identifying indicator taxa in other projects? So we haven't actually looked at our data set that way yet, so I can't speak to ours. If I'm thinking of the literature, I suppose there's two examples that come to mind, and they both come dominantly from the fungal world. And so if you get more into the plant pathology area, then I think disease incidence and the presence of a particular pathogen, there's probably a lot of evidence out there for those sorts of relationships. The other one that people have looked at to some degree, largely because there's a somewhat specific marker for it using these microbial lipids, is the presence of arbuscular mycorrhizal fungi. And sometimes higher levels of this arbuscular mycorrhizal marker are associated with, say, greater soil aggregation, for example. And so that's a group. It's not like an individual organism. And the pathogens, it's probably more species or maybe even pathovar specific. So I don't know, Chris, you've read the literature some too. Do you have any additions to that?
So as far as looking at it from an indicator taxa species, what I guess the main point that keeps coming to my mind is that we have a large amount of diversity. So we had 22,000 different OTUs, and so it brings to mind that some of these microbes may all be doing or a lot of these microbes may all be doing similar things within it, and so so picking out particular taxa or helping us find particular taxa, we can also look. I don't know where else to go.
Okay. Well, I think one thing where Chris was going with that is it may not be so much that you pick out a particular OTU or species, if you will, but we may find that there are associations of microorganisms which tend to be related with, well, from a biogeography sense, it could be to a particular suborder of soils, or from a soil health perspective, it might be to soils of a particular quality, either good or poor. And so it probably is going to be more at that association level than one single organism.
Okay, well, moving over to the chat, got a couple questions in there, and the first one's from Sanford. Cost of DNA amplicon per sample, and could you decode what an amplicon is for us, though?
Yeah. Okay. So an amplicon is basically this specific piece of DNA that you are sequencing. And so we just have a way of, in a sense, pulling and amplifying, that's why it's called an amplicon, that particular sequence out from all the organisms that contain that kind of sequence. And there are, I guess there's two main costs involved. One is the extracting of the DNA, and that does cost you typically $5 per sample. The next is kind of the sequencing portion. And that's where it's a little bit harder to come up with an estimate, because you can sequence a lot of samples all at the same time, and so it just depends how many you want to do. So I'm going to ballpark that one for you, and I could easily be off by a factor of 2 or 5 with that. But in our shop, you can probably get the sequencing part done for less than $10 a sample. So from just a simple cost perspective, you're talking maybe $15, $20 per sample, which I'm not exactly sure what the commercial labs charge for the lipid analysis, but I think it's in that ballpark. I think they're sort of like $25, $30 per sample. Let me know if that answers the question or not.
And if Sarah asks, spin on an earlier question, can these microbial compositions vary within the seasons?
Yes, they do change throughout the season. So we saw, just looking at the temporal sampling that we did in Corvallis, we saw that within one of our plots, we saw a pretty big shift between summer and winter, I guess. And in the Willamette Valley in the wintertime, we have these wet winters and dry summers. And so, which does select for different microbial groups as you move throughout the season.
But again, that was much smaller than the variation, say, I'll call it field to field or soil series to soil series. So,
all right, well, it looks like we've exhausted our questions. I want to thank you, Dave and Chris, for your time and effort to make this presentation, and thanks to all the participants for joining in. We had more than 100 people join today's webinar. The on-demand recording of this webinar will be available on our National Soil Survey Center YouTube channel within a few days, so feel free to let your colleagues know about this training opportunity. This concludes our webinar presentation.